Hello and welcome. My name is Andy Felton. I'm the Vice President of Product Management for Research and Platforms at Thermo Fisher Scientific. Today I'm going to talk about bringing the power of NGS to multiple applications within our NGS systems. If you look at the evolution of next-generation sequencing technology and its adoption cycle, we've clearly come through the innovation, the early adopter, and we're well into the early majority on our way to the level of broad adoption. The early adoption phase was really characterized by high levels of centralized testing and expertise required to operate NGS systems that typically required a very large capital investment. And the answers that those systems generated typically had turnaround time as being one of the lowest priorities. It's really about the kinds of answers that those NGS systems could generate. However, as we're moving into the broad adoption phase, we're really moving to a state where NGS technology needs to be decentralized so that more labs can adopt it. And that really means the simplicity of workflows, both the wet side and the informatics side and the analysis, and the ability to adopt flexibly many different applications. And in many of those applications, the rapid turnaround time is clearly extremely important. So for our NGS systems, we've looked very carefully at some of the barriers to adoption. And really, those that you see on the left-hand side of the slide are outlined here. So really, for most labs, it's still really too slow, requiring sometimes days and often weeks, particularly if labs are outsourcing their NGS to outside groups to get the results. The workflows are still very complex, requiring multiple instruments, that span multiple complex bioinformatic sources or resources and multiple suppliers. And that leads to costly workflows and lots of penalties to run, for example, small sample batches if you don't have a very high throughput. And in many kinds of applications, the sample is pretty limited, and that leads to quantity not sufficient failures in those results. In the world of centralized NGS testing, it's also quite slow. If you centralize that resource, that leads to long turnaround time to results, typically at least 3 days, but more like 5 days to 7 days, with fairly cumbersome workflows, including manual library prep or semi-automated at best, both pooling and sequencing across multiple instruments. And that leads to both a high cost and depending on which technologies are used, particularly for library prep, to high amounts of sample input requiring to achieve low levels of detection. Whereas what in reality most labs would require is a system that returns results quickly, that has a very simple workflow that's a low barrier to adoption by technicians, that flexibly supports different sequencing output scales, and uses a very small amount of sample to generate the results. And that's really where we've been trying to develop the NGS technology at Ion Torrent over the last 10 to 12 years. Slide here, you see the evolution of our next generation sequencing platforms. Obviously, we were not the first to introduce NGS. That was done back in 2005. But we were the first to introduce the personal and benchtop next generation sequencing system with the Ion PGM and the 314 chip that you see under that picture. We then followed that up with the introduction of our core Ion AmpliSeq targeted resequencing technology that really unlocked targeted sequencing as a simple application that can be used across many areas. We then automated those workflows through the Ion Chef, in particular, both the library, the template prep, and chip loading, and simplified the running of the sequencer with the introduction of the Ion Gene Studio and S5 systems for both flexibility and scalability of output. And that's culminated at the end of 2019, the beginning of 2020, with the first of its kind Ion Genexus system that really automates the whole process of library prep, templating, chip loading, sequencing, our analysis, and brings those results together in a single day, less than 24-hour turnaround time. So a word back on ion AmpleSeq technology. This was really a game-changing introduction in the world of targeted sequencing. It was developed out of our TACMan PCR, real-time PCR workflows, but allowed ultra high levels of multiplex. And in the case of AmpleSeq, up to 25,000 amplicons in a single reaction. That was really unprecedented. It's highly specific. Each primer is targeted for a specific part of the genome of the organism that you're interested in. And it had an incredibly simple and robust library prep workflow. It's addition only steps and fully automatable. Typically can be done from very tiny amounts down to about a nanogram of either DNA or RNA input. And that versatility has been applied in different ways. You can design your own assay. We have designed multiple different kinds of assays across many different application areas that we'll talk to in the next few slides. In particular, one of the most widely used is the ability to develop your own customized AmpleSeq panel. So we have a web designer tool at ampleseq.com that allows a user to design to any part of the human genome or a non-human genome with a single reference gene for hotspot panels. We can also design human gene expression panels or gene fusion panels. And we've also used that technology to design panels for non-human targets, including things like antimicrobial resistance panels, pan-bacterial detection, and for many targets, including things like Ebola, viruses, SARS, as well as mycobacterial like TB. So this lends applications from human inherited to oncology to infectious disease and a wide variety of other areas. If the solution required is too complex to be done through our standard AmpleSeq design tool on the web at ampleseq.com, we have a team of expert scientists known as our AmpleSeq White Glove Service who can help you design your own panel if it's, for example, a complex multi-reference infectious disease panel requiring high diversity or lots of detection or exclusion requirements. If it requires a lot of input and requirements gathering, we can do that through our AmpleSeq White Glove Service and talk to your Thermo Fisher next generation sequencing sales rep to get more information on that. In terms of the kinds of applications that ion AmpleSeq has really turned its hand to, it's incredibly wide. And you'll see here the list. We've designed panels for both gene expression, for immune repertoire analysis, as well as infectious disease, cancer research and cancer testing, cancer screening, as well as up to including the whole human exome. And it's an incredible versatile protocol that we've automated on our Ion Chef platform, where it can be automated on standard XYZ pipetting robots in a very simple fashion with as little as 15 minutes of hands-on time. And you can see the steps below, really as simple as amplify, digest, ligate, normalize the reactions or equalize, and then combine them to be ready to be sequenced. So with that targeting technology that covers such a wide range of technology and application areas, we've developed 2 generations of systems, and most recently. The first is our Ion Chef and Gene Studio S5 platform. So the Ion Chef automates the library prep, as well as the clonal amplification or templating step and the chip loading. And then the chip goes into the Ion Gene Studio S5, S5 Plus or Prime systems, depending on the compute requirements you need for sequencing. At the end of 2019, we launched the Ion Torrent NGS integrated sequencer. And late last year, we also launched the Ion Torrent NGS purification system that partners with the Genexus system to deliver an incredibly simple workflow with just 10 minutes of hands-on time for the setup of both instruments. So really incredibly simple to use. So a little more on those capabilities. The Ion Gene Studio S5 system is really the most flexible and scalable platform that we offer. You can have chip capabilities ranging from our Ion 510 chip at just 2 to 3 million reads per chip run, up to our Iron 550 chip at 100 to 130 million reads per run. And those sequencing runs take about 2.5 hours, with the whole workflow taking 24 to 48 hours maximum. With the Iron Torrent GeneXus system, that was developed with a brand new chip type called the GX series, in this case the GX5, that has 4 independent lanes. Each lane is about 12 to 15 million reads, and each lane can be run independently, with the whole chip and the sequencing reagents able to stay on the instrument for up to 2 weeks. And that allows the user who has samples coming in at random times to be able to start the instrument, run one lane, wait a few days, run a second lane or next 2 lanes, and then run the final lane up to 2 weeks later. So it really enables the user to deal with the samples that come in as they actually come in the door and turn them around with the speed that they need. To support those 2 systems, we've developed a carefully curated content list across both hemato and solid tumor oncology, as well as inherited disease testing and infectious disease testing workflows, all based around the single library technology of Iron AmpliSeq. That gives us the streamlined workflow, incredibly fast time to results, and that's supported by our end-to-end solutions, including our bioinformatics for multiple application types. I'll talk a little bit more about those on the next slide. For our Iron Gene Studio and S5 Chef customers, we have a primary application, our primary analysis tool called Torrent Suite for run planning and primary analysis. That is then paired with Iron Reporter software, which covers a wide variety of more complex secondary analysis tools, including, as you'll see here, for things like immuno-oncology, for reproductive health or metagenomics, in the world of oncology, analyzes both solid tumor, liquid biopsy, hem-onc, and gene fusion targets, and including things for analysis of the exome. So a really complicated, comprehensive, and sophisticated set of analysis tools to support the system. Within the GeneXus platform, what we've essentially done is combined the functions of both Torrent Suite and Iron Reporter, as well as our oncology tertiary analysis tools, including Oncomine Knowledge Base, to a single analysis software that encompasses both the purification system and the GeneXus integrated sequencer to really simplify the workflow even further beyond the Gene Studio S5. So in the case of the GeneXus system, you simply input the sample information for those that you're going to purify. And in the case of that platform, we can handle FFPE tissue of up to 12 samples, or whole blood for hematology applications of up to 12 samples, and for 6 samples of liquid biopsy where you're processing plasma for up to 4 to 8 mils of plasma for isolating total nucleic acid, that is both DNA and RNA. That workflow takes about 2.5 to 4.5 hours, and then it's paired with the automated library prep and sequencing and a software tool that follows those samples from the beginning to end if you're using the integrated mode, and performs the rest of the application in 14 to about 24 hours. So a whole workflow, approximately 20 minutes of setup time for both the purification and integrated sequencer, with a little bit of upfront processing time for sample types like FFPE, but a 24-hour turnaround time to result from the full system. In the case of the Gene Studio S5, we pair that with our Kingfisher purification systems that handle a wide variety of different sample input types at a wide variety of different scales and throughput levels, to our Iron Chef automated library prep, as well as templating, and then with our Gene Studio S5, Torrent Suite, and Iron Reporter software. And that's typically 24 to 48 hours of process time, with about 45 minutes of hands-on time from the chef to the Torrent Suite result. With these kinds of platforms, we enable to our customers to do some really fascinating work. And in this example, I'm showing you in November of 2020. We were November of 2021. I apologize. We had placed the Genexus integrated sequencer at the Lancet Labs in Gauteng, South Africa, and they were the first team to identify in the world the Omicron variant after noticing S gene dropout in their SARS-CoV-2 detection with those TACMAP-based PCR kits. They followed up and sequenced them and identified the specific virus mutations in less than 24 hours and alerted the South African surveillance program and the world about the Omicron variant. Another example of the use of our NGS systems is in reproductive health. In reproductive health, we have three major areas that we support: in vitro fertilization or assisted reproduction, non-invasive prenatal testing, and our carrier screening applications. And in this case, our team in Malaysia, the Alpha Fertility Center, uses the INS5 system to generate results for their IVF service to help couples in becoming pregnant. And lastly, our oncology. The Genexus system has been truly enabling in this space. The 24-hour turnaround time to result is really a huge advance for testing of oncology samples that's typically taken anywhere from 5 to 14 days. And both our colleagues, Nicola Marano in the Italian oncology up in Naples and Philip Germer at the University of Basel in Switzerland, use the Genexus system and its fully automated end-to-end workflow with very little technician time required to run the samples and generate high-quality results, even from very small amounts of material. So finally, we think the combination of our Ion Gene Studio platform with its flexibility and scalability and the ability to manage very multiple application types and the incredibly simple, integrated, automated, highly automated system with a rapid turnaround time of Genexus has really helped the mission of democratizing next-gen sequencing with over 10 years of technology application and innovations. So look forward to working with you, and thank you for your time today.